All right, last video for this particular topic, we're going to get into how cells basically control themselves in terms of division. So we'll talk about the cell cycle and interphase, mitosis, the stages of mitosis, and then some other things in terms of how we regulate this process as well and when we lose control of this process. So this is really the big thing here, is the cell cycle and understanding the basics. Now, you should have talked about this in a previous class, but so some of this should sound familiar even if you don't remember the details. So cell cycle basically is just the process, the round and round we go, the life cycle of the cell. And really this is kind of where we're going from just doing our generic daily duties as a cell to creating a second cell. And so we have three main parts to this: interphase, mitosis, and cytokinesis. Interphase is going to be the period between dividing. And so this is what we think of, we're like doing our regular business, right? Kind of just growing, we're maybe creating more cellular contents, we're duplicating things, right, that need to be duplicated. And then we get to, we create more chromosomes, right? We duplicate the DNA. We do some checks, right? Spell checks basically to make sure everything's good. And then we go into mitosis, where we divide up the genetic material, right? We divide the chromosomes. And then cytokinesis, where we actually split into two cells. It's important to note that how much time you spend in these phases and if you go through these phases or when you go through these phases can vary depending on the type of cell you are. So like something like a skin cell will go through this relatively quickly so that you can create more skin cells as you lose them, right, through in your epidermis. And you'll go through cytokinesis to do that on a regular basis. However, something like a muscle cell will go through interphase and mitosis, but it'll actually bypass cytokinesis several times before finally going through that. So there's differences in terms of how the cell cycle is run depending on what type of cell we're looking at. But in general, cells will follow these three phases, and most of their time in general will be spent in interphase. So let's take a look at interphase. Interphase has three parts, which you already saw in the picture on the previous slide, but let's take a deeper dive. So in the G1, we're going to replicate the organelles. So just kind of hang out, make organelles that we need, the Golgi, the ER, lysosomes, right, all that kind of stuff. And most cells are not necessarily dividing, and so they're just going to hang out in the G1 phase, right, just hanging out here. And so this is also like what I say is where we're just doing our normal daily duties, right, is in G1. But if we are going to divide, we can then go into S phase. In S phase, we only have one job: synthesize the new DNA, which basically means go through DNA replication. That's the only thing you need to remember for S phase. Make a copy of all the DNA. Then we go into G2. G2 is where we're going to prepare for dividing all of the genetic material in the cell. So we're going to do all of our double checks. We're going to find mistakes. We're going to fix those mistakes if we find them, so on and so forth. So G2 is very important in that it's our last chance, or one of our last chances, I should say, to find any errors and fix them before we commit to fully divide, before we're in the dividing process. Because any mistakes that are not found in time, when we go through mitosis and cytokinesis, those mistakes are now locked in. We cannot go back and fix them after the fact. Just like you would not be able to make corrections to your paper after you submit it, right, for grading, same thing. Those mistakes are locked in if they escape through from the G2 into the mitosis and into the cytokinesis phase. There's one more time where we'll get one last check partially way through mitosis, but other than that, G2 is our last chance to find any errors. So let's go into mitosis. Let's basically say everything's gone well, everything's been perfectly fine. We go into mitosis, we're ready to divide our duplicated DNA into two new sets. And so we go from interphase into prophase. So in prophase, we have condensed chromosomes. They're now visible. They look like the chromosomes we see in pictures. The nuclear membrane and nucleolus break down. That way we have exposed the chromosomes for movement. The centrioles are going to move apart to create a pole on each side of the cell. And then we're going to start sending out spindle fibers from the centrioles, and their job, these spindle fibers are, their job is to basically reach out and grab onto those chromosomes. We then go to metaphase. In metaphase, the chromosomes have been latched onto by those spindle fibers, and we're basically have been playing a game of tug-of-war between each side of the cell. Or if you look at the picture, you can kind of see that. And so you think about a game of tug-of-war where it's kind of a tie, where it's very evenly matched, right? You have that rope pulled tight from both sides, and it's really not moving in the middle. We've basically done that so that the chromosomes are stuck right in the center, right in the middle of the cell, right? Under what we call under tension. Then we get into anaphase, where the centromeres split, so we have proteins that will break down the centromere there so that we're no longer under tension. And we're splitting the chromosomes in a very specific way so that one chromatid goes one direction, the other chromatid goes the other way. And remember, the chromatids in this case are 100% identical. So we have one identical piece going one way and then another piece identical to that going the other way. This is how we're going to make sure that the cell ends up identical from start to finish, because we're making sure that we have a copy of that specific chromosome going in each direction. And then lastly, we get to telophase. Telophase is where we're actually going to get ready for cytokinesis. We're starting to split the cytoplasm. We're recreating the nuclear envelope. We're recreating the nucleolus. The chromosomes are decondensing. And so I like to think of telophase as like everything you saw in prophase, you're seeing in reverse, right? So in prophase, you saw the nuclear envelope break down. In telophase, you see the nuclear envelope build back up, right? In prophase, you saw the chromosomes condense and become visible. In telophase, you see them decondense and become harder to see, right? So think of telophase as kind of the opposite of prophase, and that makes it a little bit easier. And then lastly, if we go through cytokinesis, in our animal cells, our human cells, we would see what's called a cleavage furrow forming. This is basically like a tightening down using actin filaments. So basically actin will act like a belt. And think of if you were to tighten a belt all the way until you pinched in two. That's kind of what we're doing here. We're tightening using the actin until finally we've tightened so much that we pop into two sides. Now, sometimes tissue can become enlarged because of changes in the cell cycle. So sometimes it's hypertrophy. This is because the cells have been bigger. So we see this when someone gains like muscle mass. So if someone's muscles get bigger, muscle cells don't really replicate like that, like the way we've been talking about. So rather than make more muscle cells, the muscle cells that are already there just get larger. So when you think of someone who's getting like bigger muscles, right, they've been going to the gym, pumping iron, right, hypertrophy, right, which is sometimes the desired effect, right, for someone who's trying to have a certain appearance. Hypertrophy is just because their muscle cells have physically gotten bigger. And then hyperplasia, this is where we have more cells in terms of numbers. The cells are the same size, but now instead of like the 3 we started with here, we see 5. Right? And this can lead to what we call a benign neoplasm, so a benign tumor basically, or something that could be cancerous, like a tumor that will continue to divide and be problematic. So how do we control for this so we don't lose control over our tissue? There's a very elaborate system in place for this. It's not we're just going to be talking about this briefly. We're not going to get too crazy. But we want to kind of pinpoint some of the things that are going on here. One is what we call a checkpoint. A checkpoint is a certain spot in the cell cycle where we basically are going to regulate before you kind of check to make sure it's okay to go to the next stop, the next point. So you think about like if you were to, let's say you're driving to Mexico for vacation, right? What happens as you leave, as you cross the border, right? Or back and forth, there's a checkpoint, right? Or if you are, even better, I just moved from Iowa to California. What did I have to drive through? An agricultural checkpoint to get into the state. Even though I was still in the US, right? I drove through Arizona to get there, I still had to go through a checkpoint. So why, what's the purpose of the checkpoint? To slow things down, check to make sure everything's good before you let things go any further. We have several of these. We're going to talk about 3 of them: the G1 checkpoint, the G2 checkpoint, and the metaphase checkpoint. All of these in general are using what's called a cyclin and cyclin-dependent kinase pair that will help us determine whether or not we should get past the checkpoint. So what are we looking for? Right? What are we looking for in G1? We're looking for checking the DNA. Is the DNA damaged? Do we have the right growth factors? Did we get the signaling to even go forward? Do we have all the materials we need? Right? So this is kind of like you're checking, if you think of it like the analogy I was saying with writing a paper, this is like us, did I do the reading I needed to do? Do I have the instructions for the assignment? Right? Do I have those things that I need to get started on writing my paper? Right? Then we write a paper, right? DNA synthesis. We get to the G2 checkpoint. So I'm going to do a read through. Does my paper make sense? Right? Did I hit all the things that the instructions asked for? Right? Did I make sure to answer the questions that the prompt had? In the G2 checkpoint on the cell side, they're looking at, is the cell the correct size? Did the DNA replicate properly? Or, are there any errors in that? And then we get to the metaphase checkpoint. Now, in this case, we're going to make sure that the chromosomes have attached to the spindle correctly. So this would be, if we're using, again, the writing a paper analogy, this would be similar to you doing your last check right before you hit submit. You're double checking everything, your name's on it, right? Everything's there before you hit submit. All of these checkpoints have a place where we can stop and fix the mistake. Or if the mistake is like irreparable, there's nothing we can do to repair that damage, then we can go through what's called apoptosis, which is programmed cell death. We can stop that cell in its tracks so that we don't replicate. So we have these methods, right, of preventing damage from being passed on to the next cellular generation. So we have genes that will create things to either promote the cell cycle or slow it down. Tumor suppressor or tumor suppressor genes, tumor suppressor proteins are going to help regulate the cell cycle by slowing things down. So what they do in terms of slowing things down is they're going to stop to make sure that the genetic information is correct. They will call for repairs for DNA if there's a mutation found, and this can help prevent us from developing cancer. This works opposite of oncogenes. Oncogenes, sometimes called proto-oncogenes. Proto-oncogenes are normal, right? They're going to help speed up the cell cycle, but if they mutate, they can become oncogenes, which then are cancerous. So I like to think of tumor suppressor genes and proto-oncogenes like the brake and the gas in your car. The tumor suppressor gene is the brake pedal. The proto-oncogene is the gas pedal. You need both in order to drive a car effectively, right? But if either of those goes out, you have a problem, right? If your brake no longer functions, you can't stop, right, without crashing. And if your proto-oncogenes, if your gas pedal doesn't work, you can't go anywhere at all. You're stuck where you're at. So in this case, right, if either of these stop functioning, you can develop cancer where we no longer have control over the cell cycle for various reasons. Now, when cells divide without control, they can develop into a tumor or a neoplasm, which basically means we can be benign or malignant. So basically it's just like an extra growth. So a tumor or neoplasm is like something that it's not really supposed to be there. If it's benign, it's kind of encapsulated, right? It's kind of held in itself. So it's getting bigger, right? But it's self-contained. Whereas malignant, we think cancer, this is going to have the ability to invade other tissues all around. That's problematic because then we can have what's called metastasis, where some of these cancerous cells break off and start new tumors in other parts of the body. And so that's where you start seeing things like when people start talking about like, oh, I have stage 4 cancer or whatever, or I've metastasized cancer. That's what they're talking about. They have this growth in one spot that is no longer just held to that spot. it's now found in other parts of the body and it becomes much more difficult to treat. Now, depending on the tissue affected by cancer, we have different names. We have carcinomas. These are going to come from epithelial cells, and they're very common, especially in the skin, right? So like basal cell carcinoma. We have sarcomas. This is a cancer that comes from muscle, fat, blood, lymphatic vessels, cartilage, or bone. These are not very common at all. And then leukemia. Leukemia is going to be in the red bone marrow, so it's a cancer of the tissues that form your blood cells. Lymphoma is a specific blood cancer. This is where white blood cells are affected. And then melanoma, this is specifically of the melanocytes, right? And of the skin cancers is the most dangerous.